Okay. Good morning. First of all, let me congratulate with the organizers for this very nice Congress and to express my gratitude for inviting me here. This is CIMER, if you want to pronounce in this way, or CIMER in Italian. That is the Center of Regenerative Medicine that we have established 2-3 years ago in Rome. That is, wants to be an international center. So we have a lot of members from all over the world in this moment. So whoever of you could be interested, we will be happy to have you with us. Today, I will try to summarize some of the concepts that represent the basement of what we are doing in our labs. But as a first thing, let me make a general consideration. In present times, it's a problem with the pointer. Anyway, in present times, the structure of disease is changing worldwide. Infectious versus degenerative diseases are simply related to the wealth of a territory, of a population. So, why? I have done something? No. So in areas in which we have a low income, infectious diseases are prevailing on all the others. As soon as income increases, we have a prevalence in the degenerative diseases. And based on the World Bank, poverty is declining worldwide, and at the same time, as we know, longevity is increasing. This means that in the next decades, we have to expect a true explosion in degenerative diseases that represent a very big problem at economic, at social level, and at family level. But what are we doing against this kind of diseases? Basically, we are applying today only palliative drugs, palliative strategies, because we have no way to cure the disease, but we simply ameliorate symptoms, we delay the negative effects of the disease. So we have a number of drugs and devices that help us in this, but we have no idea about how to cure, in a final way, the disease. When we are desperate at the end, we use a radical solution that is heart transplantation. But heart transplantation is a very expensive and limited resource. Consider that every year we have no more than 6,000 heart transplantation worldwide. So it's nothing in respect to what we could need. So as soon as cell biology made robust advances approximately 15-20 years ago, someone started thinking that it could be better, instead of changing the whole organ, but to substitute only cells that were functionally inappropriate or that were dead. Today I will speak about the heart because this is my topic, but I think that some of the concepts that we are trying to study, to develop, can be applied also in other organs. And after what I heard this morning, let me make just a small comment. When we are speaking about heart disease, we are thinking to infarction. Yes, but it's not the true or the only heart disease. There is another way of heart disease that is a scattered death of the cells, like for hypertension, for example, that leads to heart failure without necessarily the infarction. What are we doing for that? Simply, we are not considering this possibility. Above all, as investigators, we are very much attracted by something that provokes pain and a very acute condition also in the family. But we are not considering that there is a hidden enemy that works against you day by day. So, as soon as this idea was established, imagined, there were some questions that I have summarized in these three. Excuse me. Which cell type should be considered optimal? It seems to me that we are at the same place today after 20 years, okay? Which protocol should be applied ex vivo? No one discussed about this second question. I didn't hear anything this morning, and even in other situations. Which implantation procedure could be the most efficient? Nobody knows, okay? So I will try to make some comments on these three items, on these three issues, for what I can. In search of the optimal cell type. Sanjeev already showed that there is a plethora of cells that have been candidated and that we can summarize in this: bone-derived, adipose or cardiac-derived, or pluripotent stem cells, or someone is trying to find a way to induce cardiomyocyte proliferation in situ. But less than 1 year ago, Circulation published this consensus report where all types of cells candidated were reviewed in depth, and they established that no one of them had the prowess to satisfy our expectations. Plus, 2 months ago, Nature published this review on the clinical trials. And they concluded that no one of the clinical trials at the moment guarantees the use in the clinical setting. So this morning we heard many other things. It means that we need to go back to cells to find new markers, maybe to find new subpopulation, to have new, very innovative ideas on how to find the optimal cell type. Delivering regenerative signals. This is what, in general, we are doing. We isolate some stem progenitor cells. We implant them to obtain the repair of the bioarchitecture and so of the function. Since a few cells, a meager number of cells are homing for what we know today, someone imagined that it's a secretome more than the cell itself to give some amelioration. Today, we will be concentrated on the implant, not on the secretome. So, until now, cells have been delivered by injection. Why? Injection requires no skills. It's inexpensive. You can do whatever you want. But the results, as I have already said, have been not satisfying because of maybe a unsystematic approach, or because we do not know anything about how cells behave as soon as inside the recipient tissue. Nobody studies these aspects. How cell adapts to the whatever kind of cells as soon as you inject or you administer that. Finally, unstandardized procedure. Every lab dreams to win the Nobel Prize. So it's impossible at the moment to imagine, also in Cleveland, they are thinking to the Nobel Prize, I know. It's impossible to think to have a standardized procedure. I cannot be treated in Cairo in a way, in Melbourne in another way, et cetera, et cetera.